by everybody in science because it's pretty amazing. One of the things I found in the very beginning of it is, so there were 7,000 described diseases. How many of those 7,000 have a treatment? Any guesses?
Have a treatment?
A treatment. At least one treatment for it.
I bet it's pretty high, half. 2,000.
500. I was shocked.
Wow.
500. And some of it is due to the science that we're going to talk about. So, all right.
So just for
Yeah, okay, great, thank you. Okay, so again, this was the book, Rigor Mortis, and one of the things that I really liked in it was, well, many things, but something I took away was this, provanda e riprovanda. Does anybody know what that means in Italian? Provanda e riprovanda.
try and try again.
Test and retest, yes, exactly. And this was from just after Galileo died. Around 1642, there was a group of experimentalists in Italy, and they began publishing their work, and their motto was test and test again. And I think that certainly holds true to what we're doing today. So scientific rigor and reproducibility, easy definitions, right? Okay, so I looked them up. So NIH has a definition of scientific rigor: the smooth application of the scientific method to ensure robust and unbiased experimental design, methodology, analysis, interpretation, and reporting of results. This includes full transparency in reporting experimental details so that others may reproduce and extend the findings. Okay, that sounded pretty good. But then you get to reproducibility, and it's a different story. So this was from the NSF subcommittee on replicability in science. So reproducibility, according to them, refers to the ability of a researcher to duplicate the results of a prior study using the same materials as were used by the original investigator. Makes sense, or is it? So then I came across this paper. What does research reproducibility mean? And so this was from a group out in California, Steve Goodman and John Ioannidis, and they've been spending a lot of time thinking about the problem of reproducibility in science. And a lot of this stems from the issues with preclinical research then not leading to treatments down the road. So they actually proposed a new lexicon for research reproducibility, and they've broken it down into three. Methods reproducibility, which captures the original meaning of the word, and they refer to this as the ability to implement as exactly as possible the experimental and computational procedures with the same data and tools to obtain the same results. They then coined results reproducibility, which was previously described as replication. So that's one of the issues. There are a lot of synonyms that one can use. And this is the production of corroborating results in a new study having followed the same experimental methods. And then their third group is inferential reproducibility, the making of knowledge claims of similar strength from a study, replication, or reanalysis. And then they put this chart here where they looked under Scopus, and they searched for the words research reproducibility, reproducibility research, reproducibility results, on and on in abstracts and titles of papers, and they found this curve for many different disciplines of science, how since 1970 up through about 2012, it has really spiked in the last 10, 15 years as people begin to think more and more about these issues. So if you're a fan of Sherlock Holmes, I am, and this was a recent book by Maria Konnikova called Mastermind, Discovering the Sherlock Holmes in All of Us. And this was a quote I took from it. And she says, begin with a healthy dose of skepticism instead of a credulity that is in your mind's natural state. While Watson is quick to jump to conclusions, Holmes forces himself to reserve judgment until he has all the facts, a more effective path. Once people reach a theory, they tend to single-mindedly focus on confirming it, regardless of the facts, thus inviting error. Holmes knows that if he comes too quickly to a judgment, he'll miss much of the evidence against it and pay more attention to the elements that are in its favor. And I think that's a trap that we as scientists tend to fall into, is that we have our own pet hypotheses, and we tend to be blinded sometimes to results that don't confirm or adhere to those. I think that what she said that Holmes believed was very accurate. So here are some thoughts on rigor and reproducibility that I came up with. And I thought that it could drive the discussion once this presentation is done. So a lot of this began with clinical trials. So it was a paper by Ed Begley a number of years back, and he was working for Amgen at the time. And he decided to go into the literature and look at 65 what he called paradigm shifting papers that were preclinical research. And he said, let's see how many of those we can reproduce here at Amgen. And the results were 11% could be reproduced, either at Amgen itself or asking the original investigators to go back and redo their work, which they would pay for, but to see if they could reproduce it themselves. In almost every instance, they couldn't. And so he published this, and it caused quite a bit of controversy. A lot of this can be attributed to the batch effect. And we can talk about the batch effect again during the discussion. Clinical diagnostics. So in our lab, in the Microscopy Imaging Center, we also do some clinical diagnostics. So we are licensed by the College of American Pathologists, which means that we have to have SOPMs. So we get inspected on a biannual basis, and all of our procedures are checked. And so at least in the diagnostic world, the rigor and the reproducibility that we are subjected to are very intense. That's not often the case in basic research. So if we think about basic research, let's think about those who are doing studies with animals. What are the number of animals that you need for a study? Power calculations will oftentimes give you that number, but unfortunately, oftentimes it's driven by how much money is available to house and maintain the animals. So this was a group called the ALS Therapy Development Institute down in Boston, and as the name implies, they're looking at treatments for ALS. So they focused this institute specifically on one disease. They did a very similar thing like Begley. So they went back and they looked at all the preclinical papers, and they wanted to know, why are there no successful drugs for ALS at this point? And when they went back and looked at the preclinical data from the animal studies, they were shocked at the lack of rigor in those studies. Some of the papers published experimental results on these drug treatments using animal groups of 4 animals. So this institute is now redoing these studies, and they're using 32 animals per group. Who can afford to do that in academia? They get no funding from NIH because they said NIH will never fund them to do this. And if they did, they couldn't afford it anyway. So they rely on donors, exclusively on donors, but they're redoing all the studies using very large groups of animals. Blinded analysis, they found that in many instances, the investigators were not blinded to the experimental groups. Statistics. So, I mean, this is something that has become quite an issue with respect to p-hacking and hacking. Is everyone familiar with these terms, p-hacking? So p-hacking, so the p-value, so p-hacking is basically running the data every which way you can until you reach 0.05 and then you stop, and that's what you present, okay? Hacking is the hypothesis after the results are known. So you look at all your data and you say, oh, wow, here's something significant, and you go back and change the hypothesis. So this is something that's quite an issue today as well. Good laboratory practices, our laboratories using them. I mean, industry certainly does, but research labs not as often. There has been a call for something which is being referred to as good institutional practices, where an institution actually mandates these are the types of rigor and reproducibility that we expect from the institute. Reagents. So this is another big issue, something that I think about a lot in the facility, because in my own research, as well as people who come in and use our facility, as well as being a journal editor, the validity of antibodies. So a lot of these studies which fell down here were due to the batch effect. The majority were actually antibodies that weren't specific, that ended up causing a lot of the issues. Validated cell lines. How many here who are using cell lines have actually validated them to make sure they're the cell lines that you think they are? Maybe everyone's doing it, but an awful lot of papers have been published. The most egregious example was a cell line which was purported to be a breast cancer cell line and was used in thousands of papers, and when someone finally took the time to validate it, it was a melanoma cell line. It was cancer, but nevertheless, it wasn't breast cancer, which everyone thought they were working with. So there is also an initiative now called the RRIDs, Resource Identifiers. Have people heard of those? We'll get to that in just a moment as well. And then publishing. So journals have a role to play in this as well. And there are predatory journals, and as Professor Irvin just mentioned, impact factor plays a role in some of this. Okay. So Bagley wrote another commentary based upon his paper, and he said these are questions researchers should ask regarding experimental reproducibility. So whenever you're reading a paper, you should think about, were the experiments performed in a blinded manner? Were the experiments repeated multiple times? Were all of the results presented, not just cherry-picked showing the best results? Were positive and negative controls included? Were valid and validated reagents used? And were appropriate statistical tests used? I think we as scientists all assume that the answer to these are all yes. But in fact, if we really get into the weeds, we find that oftentimes that's not the case. So NIH has taken a good look at this, and this is from the FASA Washington update from October of this year. So it says NIH, to enhance rigor and reproducibility of basic biomedical research, were discussed in a meeting that they held there. Noting that NIH efforts alone will not affect real change, the deputy director stressed the need for all stakeholders, individual researchers, professional societies, research institutions, and scientific journals, to engage in efforts to enhance reproducibility and transparency of scientific studies. Building on this idea, Tim Errington at the Metascience Activity Center for Open Science discussed some of the common roadblocks and challenges when reproducing published works. In addition to well-cited examples of small sample size or inappropriate use of statistical tests, the ability to reproduce an experiment can be affected by lack of access to key reagents or incomplete documentation of research protocols. So again, how many of you have tried to repeat an experiment that you've seen published and have failed to do so, and in many instances because the details that you need to redo that experiment aren't presented? You may or may not be aware that NIH actually has a site devoted to rigor and reproducibility. If you could go on there, it's very interesting. There's a lot of information. A lot of it is for preclinical science, but they do talk about two of the cornerstones of science advancing our rigor rate in redesigning and performing scientific research and the ability to reproduce biomedical research findings. The application of rigor ensures robust and unbiased experimental design, methodology, analysis, interpretation, and reporting of results. So it's a nice website if you have a chance to go on and move around on it and see what they have to say. But one of the things that NIH has implemented that you're probably aware of is that since January of 2016, every grant now needs to follow these new guidelines. And one of them that I want to highlight here is the second one, which is design, rigorous experimental design for robust and unbiased results. But then a new attachment for authentication of key biological resources. So now you actually need to provide authentication of your cell lines and the specificity of antibodies. So NIH is taking a long, hard look at this. They do have a nice resource chart that you can download And they do, again, talk about scientific rigor here. They give you the definition. And then again, under authentication, key biological reagents, and they tell you which ones you need to authenticate and how that needs to be done. So with respect to authenticating reagents, this is that resource identification initiative. And this is from a group that's looking to code all research reagents with a specific tag, much like a fingerprint. And so you could then look up a reagent that you're using. For instance, in this one, they've looked up a polyclonal GFAP from Invitrogen, an antibody, and it takes you into the website, and then it gives you the catalog number, but it gives you then the RRID number that was assigned to it. So in using these identification numbers in a manuscript, if someone has used an antibody and lists what this RRID number is, you can then look that up and know exactly which antibody they use, because that's one of the issues we tend to run into quite often with antibodies is the name of the antibody will be listed, but the exact clone or where it was from isn't there. So this is an initiative that some journals have started to mandate in manuscripts that are submitted that you use RRIDs for most of your reagents. So that's, again, journals are starting to do that, and Journals Uniting for Reproducibility. So this is from a Nature editorial from 2014, and they've come up with some guidelines which they hope will encourage journals to use a checklist to ensure the reporting of important experimental parameters. And they list some of these on here. They're ones that we've talked about and gone over. But this is one, for instance, from Nature Methods that I downloaded. And so once the paper is accepted, then there are all of these checkboxes that need to be filled in, as well as further information. And the one that interested me quite a bit was the antibodies. Describe the antibodies used and how they were validated for use in the system under study. So then I went over and looked and saw, well, how did they do it? So here's an antibody that they use, TTF1, polyclonal. And then the validation is Santa Cruz recommended this antibody for immunohistochemistry studies, observing nuclear localization in formalin-fixed paraffin-embedded lung tissue. Does that validate the antibody? No. Absolutely not. They're relying on a manufacturer to say, oh, you can use this in lung tissue, which was not what this paper was about anyway. So, I mean, this is a good beginning, but I think that this probably should, in my opinion, should be given to the reviewers during the actual review of the manuscript. This is submitted after it's accepted. So to me, it's a good start, but it needs to be policed somehow. So in my role as editor, I rewrote this method section for our journal, and this is for imaging, because imaging experiments are oftentimes difficult to reproduce as well. So now if somebody submits a manuscript where they did microscopy, they need to list the type and manufacture of the microscope, the objective lens specifications, type and manufacture of camera, acquisition software, the acquisition information, including all of this information, and for fluorescence, all of these settings. These are typically things that don't show up in papers, but we decided that they would be of importance, and we need to know them for reproducibility. This is also from NIH, from this open mic from November 8th, and continuing steps to ensuring credibility of NIH research. So this one is now about journals, selecting journals with credible practices. So the scientific community is paying increasing attention to the quality practices of journals and publishers. NIH recently released this guide notice to encourage authors to publish in journals that do not undermine the credibility, impact, and accuracy of their research findings. This notice aims to raise awareness about practices like charging publication fees without notice, lacking transparency in publication procedures, misrepresenting editorial boards, and/or using suspicious peer review. If you're like me, you probably receive multiple emails per day asking you to submit a manuscript, typically to a journal which has nothing to do with the type of research that you do. These are the predatory journals. So the NIH is saying that all in all, to help convey the credibility of your work, be careful where you publish. We hope that our community publishes only in journals that do what they say they'll do. If the rigor of your work is clearly conveyed in writing and published in journals that maintain high quality standards, then your work will be viewed with respect. By taking these approaches, we can continue ensuring the credibility and trustworthiness of the biomedical and behavioral research findings resulting from public support. And obviously that's a very large issue because Congress has stepped in on this when they've heard about the reproducibility or lack of reproducibility issues. And so we do need to continue to work towards that. So this was just one, I don't know if you've seen this one, but this was an actual publication. So if you're a Seinfeld fan, you may know this episode on uromycetitis, which was a made-up term, and it was basically an episode in which Jerry was in the parking garage, and basically they got lost, and he was there for a while, and he felt the urge that he had to go, and there was no restroom, and so he went, and the security guard happened to see him and took him away, and he said, I have uromycetitis, and if I don't go when I need to, I could die. It will poison me. And so there was a journal, a scientific writer, and he has a really great name. His name is John McCool, and it's a real name. And he was tired of receiving these invitations every day to publish, and so he wrote this paper for this journal, Urology and Nephrology Open Access, which is a completely fictitious case report about Jerry Seinfeld. And the paper was accepted after 2 days, and it was published. This is their website, and it was published until notification was made about this, and then they quickly took it down. But you can still find it if you look at it. It's the author was Martin Van Nostrand, which was an alter ego of Kramer, when he wanted to be the author, and all of the names were completely fictitious, the references, everything was fictitious, and yet it was accepted within 3 days. So some potential factors influencing rigor and reproducibility. So these are a few I thought about, and I thought this would lead us into a segue to the discussion. So some of the things I was thinking about, pressure to publish in high impact journals, which can likely or can affect your promotion and tenure as a faculty member. Pressure to obtain extramural grant support, pressure from superiors to publish too soon, journals seeking to publish attention-grabbing studies, scientific ego. Again, from the book, In the Rat Race, Harris says, referring to scientists, they need to get their next grant, publish their next paper, receive credit for everything they do. They have stepped into a world where career motivations discourage best scientific practices. Okay, so I tried to keep it within 20 minutes. I think I did because we got a little bit later start. So I thought that this would allow us to have a discussion about this. And again, there are a lot of people here I think can speak well about it. And so I'm just going to open it up for discussion.
Questions?
Questions, but this, I hope this discussion. Yes, thank
Doug,
you.
I'll say it's like reporting your weight on your driver's license application or doing your taxes yourself. We all fool ourselves into thinking we're doing this right, and very few of us probably are honest. I think that in order to change, this needs to happen with current investigators who make funding decisions to begin with. So people who sit on study section, I know when I look at these outrageous grant proposals that say they're going to cure everything under the sun for $250,000 a year, I realize it's impossible for them to do what they say they're going to do. Even in their application where they have signed and said that this is acting somewhat akin to a contract, there's no way they're going to be able to do what they say they're going to do with the money. If we impose upon ourselves what we say we're going to do in our validation section, it's going to take more time, it's going to take more personnel time, it's going to take more reagents. I think we need to think realistically about what people are proposing in their grant applications to do. And I don't like to say lowered expectations, but be more realistic. about what we expect people to do in 5 years, in 4 years, with $250,000 a year. And it starts at the review of applications. And it's really coming upon ourselves to try to change the mindset, because the public who pays for our work in most cases thinks we're doing all of this right. And in reality, I say we're not.
They do, you're right. There has been a lot of trust in science, and that's being weakened by a lot of issues that have shown up in the general press about failures of many of these studies. I mean, patients are waiting for cures, and they're not forthcoming in many instances. And the question is why? And is some of it due to rigor and reproducibility? Leonard.
Yeah, just to add on to what Matt said here with respect to grants, that over the 17 years I've been here, grants are still the same nominal amount of money attached to them. But we all know that costs have gone up, in particular costs for personnel. And so the amount of money we can actually spend on doing the real research has actually shrunk over the years per grant.
True.
So that doesn't, I mean, not to put the blame on anybody else, but the expectations has to be, in line with what amount of money you have available to this.
Well, and many more investigators vying for a smaller piece of, smaller pie. There's just not as much money for the number of investigators who are applying for it. And, the other issue is, I think, that the problem is that I saw as I started reading more deeply into this is that there are so many layers to this that need to be thought about and changed. And maybe Professor Irvin, as our Associate Dean of Faculty Affairs, can speak about it. I mean, one of the issues that institutes have is looking at promotion and tenure, and where are you publishing your papers? You talked about when John Loesch was here, who's actually quoted in this book a couple of times, but the CNS papers, and how many people are vying to get their results published in a journal like that, because that's what they need to move on along the career path.
The scandal there is the high correlation between retractions and impact factors.
Yeah.
There's several studies that have looked at that, and all of us have had that experience where we look at a paper in one of the CNS journals and we say, really? Or many of, there have been some of us have tried some of these things that didn't seem to work in our hands.
Yeah.
And then this very curious phenomena where there are certain people in our fields that where everything they put their hands on comes up roses, everything. So we all get lucky at times, I suppose, but I certainly do, and I certainly haven't. And experiments that don't work, I mean, most of my experiments never work the first time. So the ones that do, those are the ones that get me nervous.
Yeah.
It's like I did something wrong.
Yeah.
There's a whole impact of the, 4 studies that get published that really happens as far as patient care goes. So a lot of the core studies get into the literature, and then an expert panel decides, yes, that's a good treatment, and next thing you know, it's in the supposedly evidence-based guidelines that, that physicians and others are trying to follow. And so it's not just patients in a way that are waiting for treatments, it's the doctors are supposed to be and are trying to, for the most part, apply evidence-based medicine in their practice, and there are, stuff that just does not work, not even doesn't work in the lab, and it definitely doesn't work in real life in practice. So it's a really serious problem. John Ioannidis, that you mentioned in the beginning, has sort of made his kind of life's work into, on the one hand, I guess appreciating the idea of evidence-based medicine, but at the same time, kind of saying a lot of the stuff that you're looking at as evidence is really is either slim or not there. So I mean, this has a huge amount of impact in real
Well,
life.
in clinical research, what you notice is that a single site study, so you do a study where
Right.
I recruit 10 patients and I do something like that. And then you roll that out as a multi-center trial, and it just, the effect size goes down smaller and smaller. The group that I work with, we've made our life's work in publishing negative trials, and it's very tough to get these published. And the two that I'm going to talk about one of these days when I get a chance were based on really strong preclinical work done on single-site pilot study that looked very good, and then we rolled them out in a 19 center studies, total negative results, and they just baffle me. I don't understand why that is. I guess it has to get back to the biases that Doug was talking about.
Well, I mean, some of it is even, more difficult than that. So one of the studies mentioned in this book, so there was a group in Boston and a group in California working on some cancer cells, and they couldn't correlate their results. Doing the same work, they couldn't figure it out. They went back and forth. They checked everything, couldn't figure it out. So finally, they said, we have to get together at the bench and do this side by side. So they sat down, two postdocs side by side, and they began doing their work. And they got to a step with the cells where the cells needed to be mixed. And in California, they put them in a beaker with a spin bar. And in Boston, they put them on a rotator, rocker. That was the difference.
That's systematic.
I'm saying that was what caused the difference in their results. So when you're talking about these multi-center trials, unless everyone is doing it exactly the same way,
they aren't.
That's the point.
And there's all kinds of problems. I mean, there's a friend of mine, one of the guys I trained in Denver, went to work for one of the drug companies. We got talking about spirometry, which is my thing. He said, you have no idea how bad the data we get from our different clinical sites, which are mostly docs in practice trying to make some bucks on this. And it's you look at it, it's really poor. And they're just grinding it out to get it done and get paid.
You have a comment? So, no.
Well, I have a question. I'd like an opinion on, I'd like a consult. So imagine a situation where you find an error in someone else's published work. We're all talking about the challenges of policing ourselves. I think we all we agree that it's hard to do good science, and it's important that we work together across sites to do good science. Sometime I have an example myself, but certainly we've read recently about cases where one researcher will bring to someone else, to an author's attention, that there's an error, and the author doesn't agree that there's an error, and then you've got this position of of course the author didn't make an error, and of course the error is obvious to the person reading the published paper. Where can we go from there to collaboratively make corrections to fix the errors that are in the literature? Like, you've published negative trials. Do you ever do that with the authors of the original trials? Do you invite them to participate?
We were the original trials, and all the work was done by one of the colleagues who actually designed the trial.
Gotcha.
So, but we could ask a journal editor. I mean, if let's say you.
I mean, I can be very specific. There's a paper that I've brought to the attention of peers here to make sure that I'm correct.
Yeah.
Is there an error in this analysis? And the answer is yes, there is an error in the analysis of this published paper, published many, many years ago.
Like how many years?
2000, I think.
Oh
I
my
mean, like
God,
a long time
that's really good.
2,000 years ago? Wow.
Wow.
Was it on a tablet?
I wasn't quite as white back then.
So there's a paper published around the year 2000 that has an error in it that is now starting to sort of proliferate into other contexts. And I have been
Well,
dragging my...
The journal still exists?
The journal still exists, but they've never, they don't have any examples of corrections or arguments about things that have been published. Like, it's just not a thing.
Well, you can
that
bring
they-
it to the attention of the current editors, which are probably not the same people, although
Correct.
they may. You could certainly write a letter to the editor.
There's a lot of interest in the publication world now on that question, because really our choice now is to retract something, which is often involuntary, and that carries a scarlet letter with it that nobody.
Oh, boy.
So journal editors are thinking about different degrees of correction that an author themselves can make to their articles. We had one, I'll give you one example. We're using PubMed Commons to do this on my own articles. We published one about 10 years ago. We published an observation. The methods have gotten better, and we realized that observation was wrong. So I went back onto our original paper and I said, the conclusions all stand here, but this one figure, and I referred to the new paper where we published the improved. So there are ways to do it, authors themselves. You're talking about if you've discovered something that somebody else has done. And I think that the evidence would show the most effective way right now to do that is either via PubMed Commons or in a blog post. I mean, most of the errors in the literature that have been flagged through blog posts. That science paper on arsenic-based DNA, that was actually proven to be wrong within 2 days by discussions on blog posts, and the community immediately knew it was wrong, and that led to the ultimate retraction. So that's not a great way to do it, but for your own work, I think there are ways to do it, but they need to be refined so that they don't carry a stigma, because actually we should be rewarded for correcting our own work, right? But right now, there is a scarlet letter aspect to it. But correcting other people's work, I just think it's so important, because as you said, the mistake gets permeated, and some little subset of people know it's wrong and they don't base their experiments on that. But others who don't know, it can lead to lots of, Richard Harris talks about that.
Right, and we find that the citations keep citing that original paper, even though it has been, say, debunked or proven wrong. The Begley paper, where they could only reproduce 11% of all those preclinical studies, not one of those studies has been retracted, even though it's been proven that it's erroneous. Not one. They still stand in the literature, and they're still cited. Now, to get to your point, Gary, for your own correction, one of the proposals out there is that, I think in Vermont, I don't know if we still have it. We used to have a tax-free day, Saturday or something like that in the spring. They're proposing a retraction day where people can go in, no questions asked, and pull one of their papers or pull a paper out and say, nope, I'm not even going to discuss it, and it just kind of goes away. To take away that stigma of actually having to go through the entire process, that you can just go in and remove a paper from the literature, because Gary's exactly right. People don't want to do that once it's out there. But you have a much more serious issue, which is you're finding an error in someone else's work. And how do you approach that?
There's a pretty famous paper from about 5 years back in which researchers compared the leukocyte response to sepsis in humans and in appropriately validated mouse models, and found that transcriptomic analysis found that there was no correlation between human and mice. It then called into question all immunologic research being done in mouse models. People looked at that and said that that's just not acceptable, and another group used the same data that had been deposited into public repositories and did an alternative statistical methods analysis and found that there was almost 100% correlation, and published that as a new paper. So instead of going back to the editor and saying there was a fault in the statistical analysis, they actually ran the experiments themselves. They used validated methods. And so I would say it might be incumbent upon you to try to publish your analysis of their system, your data, whatever it is, in another journal. and have it be 17 years more recent and see if that flies, because that was really important to mouse immunobiology in general, to have someone come out and sort of alternative approach is actually more valid.
That actually is where the blog refutations have been most effective. And there's a guy named Yoav Gilad who's done that for a couple of key papers from Eric Lander, for example, that have been just stupidly wrong. And he's gone in and actually taken the data and in his blog post reanalyze it. So a key part of that is data accessibility. And that's something that some journals are sort of boldly pushing towards, but other journals are resisting because of Doug's fifth bullet point, their scientific egos, that you really shouldn't be able to publish your results without publishing the data. So exactly that kind of thing can go on. And that's a pretty non-threatening way to do it. If you take this, well, it's threatening, but at least you have the data, you're backing up your contentions with the data. But the data have to be available in order to do that.
I think one of the things that's not sort of part of the conversation quite yet is, not part of this conversation quite yet, is the relationships in the scientific community. So I know the person who wrote this paper, and I've told her I think it's wrong, and she's told me that she thinks that it's not wrong in conversation before I did my research training. So at that time I wasn't able to say, like, I wasn't able to really show her why I thought it was wrong. Now the data in the paper would let me redo another analysis and demonstrate where the mistake is. And it's this, like, okay, do I go back to the original journal? Do I try and like co-author a paper with this person to say, like, this is where the mistake is? And those communications and like how there's like no guidebook for, there's no like rules of etiquette for
There's
that I have
there's
found.
common sense.
I have none of that. It's just
Okay, so
social common sense is not my thing. I do not know how it works.
well, that's okay.
I want a guideline.
I think it's important to understand. You know, we all have our weaknesses. So yeah, I mean, it would be, I guess, as a colleague, you'd say, well, I could give her a chance. Here's my data. We could co-author something together. She says no, then you gave her a choice anyway. And then I like the idea of publishing a paper about it, frankly, whether it's in that journal or something similar. You know,
It's really hard though when something's published already to actually, the burden of proof is so much harder to argue against something that's published, because editors don't want to admit they did anything wrong either. So I agree that's the most rigorous way to do it, but that's not a good way for you to spend your time, because it's gonna take 3 or 4 times the amount of effort to get something to a publishable state.
I was about to say that the guy who did my PhD was doing that. There was a study done in UCSF that was not only in the literature, it was in the textbooks. And he spent a fair bit of his career showing it was dead wrong. But the number of studies we had to do, different animals in different settings, to consistently prove that it was wrong.
Could I say one more thing and then I'll shut up?
Please.
I actually think that a big part of the problem here is just our culture. And if we had a really robust culture of post-publication commentary, and we didn't have this idea that because you published it in Journal of Immunology or Cell, it's correct, right? If we all sort of acknowledge that's not true, and we had this post-publication culture of commentary and realized that results evolve and ideas change over time, there wouldn't be this stigma associated with being wrong. And for some reason, biologists, they just can't handle that. I mean, my daughter's a graduate student in philosophy. You should see how they rip each other apart. And it's just part of their culture. It's just part of being an academic and being able to defend yourself and being able to give and take. But somehow we're very tender flowers in biology. We can't handle that. And it would be really good for science if we could. And if we realize that every study, you know, that's just a snapshot in time, and every bit of science evolves. And if we had a system to sort of encourage that evolution, I think we'd be better off. That doesn't address your question, how do you deal with it
The
now?
discipline I'm in was in, we trained in was vicious. we attack each other like crazy, but it's not the man.
But I think that, well, I agree 100%. It happens before publication. These are the kind of conversations that need to happen at Gordon conferences and Keystone symposia and big international meetings where you actually can confront people in an open arena and not fight, but talk as scientists. And we have lost that. There was a person who was on my dissertation committee who has written a lot about this for the scientist. And it's just, the culture has changed in the time that the 20 years since I earned my PhD. We are a lot more quiet.
Defensive.
Are scared to offend? Is that what it is? You go to meetings and people don't ask the type of questions that
The
they
change
once
I've
did.
seen at the meetings I've gone to in 40 years is really stunning. It's gone from.
And people don't talk about their recent work either. It's only published work at some meetings. It's a shame.
Used to be lineups to speak, and now it's hard to get anybody to ask it.
I think it comes down to the bottom line, and that's money. Because again, you know, when grants are down to everybody gets a score, pops a champagne cork, despite it's not fundable. It's kind of ridiculous. In fact, most grants that get the score probably would been funded, 20, 25 years ago. And now we're down to some sort of lottery to win a grant. You need to be friends with everybody in that committee. You can't disturb any relationship. You can't run the risk of having anything happening to your grant. And I think that's part of what is driving us to be very protective about our thing. So the scientific ego is not a healthy ego anymore.
I mean, I think clearly this discussion is showing that, as it's mentioned in this book and in countless articles, that it's broken. The scientific research endeavor, as we do it, is kind of broken for some of the reasons that you mentioned right there. Is that it's not. I mean, when I started out as a student, and as Charlie mentioned, things were much different, and it's changed. It's changed dramatically, and you're right.
I can add another thing to this, and it's still a bit of a difference between Europe and the United States, and that the lack of structural money in our system here in the United States, meaning that very few people actually have a secure basis on which they can do what they do in philosophy. But they're probably less dependent on grants in the way we are in biology and medicine. At least in Northern Europe, there's still some structural money in different research institutes. If you get a position there as a lecturer, professor, whatever, you have some funding that you can live off of and buy your mice or cells and reagents, whatever you like. But that is sort of dwindling down there too.
I could be wrong, but we certainly have seen some pretty egregious cases come out of.
I'm not saying it's not foolproof, of course not, but
the literature is very clear on all of this, and that is that it's the training environment, and I think one of the reasons we're doing, so I have to make a plug, one of the reasons we're doing these is to actually have open discussions about it and to provide people who we train with viewpoint. Because in the end of the day, it's your reputation. And your reputation is everything, and once you lose that, as I've seen in many cases, you're not getting it back.
If you think about what incentivizes, just as I was just saying, those first bullet points, they're all just about incentives, right? And our incentive, and I think that quote there is a great quote. Just the way science is incentivized now promotes bad behavior.
It is. All right, guys, well, thanks for coming.
Sorry, go ahead. Yeah.
Well, I was just gonna, I mean, it's to that point where if you're newer in the game, you have to get a grant, you got to have your pilot funding, and your pilot projects are not really strong studies, but if you want to keep moving on the track, you got to publish them, right? And so you just kind of perpetuate this rat race because you're publishing these premature studies because you're forced to do that in order to show you're productive. But the other thing is, I think our system, the structure of our system promotes this, where if I want to not be on completely soft money, I have to be in a tenure track position and a PI, when, you know, so we have all these people that have to be independent investigators. And there's systems with postdocs, but once you're out of the postdoc, then you need to be an assistant, you know, I mean, an independent, or you're going to go into a very strong lab that will continually fund you, but it's still soft money. So we have this system that promotes all these people that don't necessarily need to be leading studies when it could be a bigger team approach.
Well, that's being more and more recognized. The NIH, particularly NCI, is pushing that very hard, the idea of team science. I went to one of their team science things, and they were saying how the universities you guys have to change your promotion criteria. And I went, oh yeah, good luck with that. Tell Harvard how to change, yeah, right. And here we're actually pretty good at that. We use what I've come to know is a holistic, that's when you work holistic, RPT review. We recognize people's contributions to the greater good. In fact, I can't think of a better example of that than my esteemed colleague to my left. And I think we do, and there's other places, Case Western, a couple of others, they're leading the charge. Not everybody is going to be a PI, not because they don't want to or they can't or whatever. We do work as teams. Certainly here at this institution, we've made changes to that, our policies to make sure that continues to be the case. Tenure is a dream because most places are going away from it. And most of us with tenure believe in post-tenure review. So every 5 years, I should get heavily reviewed as to whether or not I still deserve that honorific. Because tenure is not about wealth, you know, scientific welfare. It's about protecting the ability to say that, you know, the Earth is flat. Flat. You missed the boat on that one. So I mean,
Missed the boat on a lot of them.
yeah, I can tell you some heinous examples of that where the institution, in one case, came down on a person just because of the money. It was money flow and influence within the university system. So it's, you know, it's not perfect, and that's why we have to have these conversations. And of course, we're all PhDs are on that road, and so we have degrees in philosophy. We practice at the top of our life. All right, thanks for coming. Happy holidays, and hopefully we have another series, 2 series. Hey, how are you?
Dr. Stern. Quick,
Yes.
are you sending me on?
Good to know.
I'm Nancy Galvin. I have a grant with
I love it. All right. Good job, Douglas.
Nice to meet you. I moved. I tried this out.
You're out of your brains.
Just turning around and Nancy there was like, no, here's idea. Yeah, I'm out of it.
I'm very curious about the device on the
That was great.
table.
This is fun.
Yeah.
We're gonna get more people involved.
So that was for recording of the question.
Yeah, we're kind of hesitating whether or not we should record the Q&A or if we just do it all. I'm not sure, so.
Statement or somebody, that, and I'm really, training.
On our website,
You can't,
And I think
I'm not telling us what I'm supposed to do.
so I'll end up posting. I was actually talking to Jason on this.
That's one of the reasons
I don't know who Jason is. That's okay.
He was the guy that was setting all this up.
We have to introduce it, we're trying to introduce it into our seminars.
So this is what,
Because a lot of it's about.
this is the page that I manage. So we'll just put it up here at some point.
One of our thesis reviews.
Oh wait, I did put it up.
Are you in here?
we're talking
That's
about
awesome.
blinding.
So you can listen to Matt Pointer's, Dr. Pointer, who's back there, his one on NIH Bioswitch. So then what I'll do is I'll take this down, I'll put the Rigor Reproducibility one up there once I get it from Jason, the IT guy, and I'll put it up there. So this is just a good site. This is, since you've been to so many other things, but kind of a high, like actually, like review with Dr. Urban, here's there's the repoints. Okay, so you can go into the different ones. So this is the faculty development one that we were just on. Okay, and then this is the reappointment, promotion, and tenure. So there's a promotion readiness, but your new CV for RPT. This is where that is.
This is where I'm like not making eye contact because I have not made any progress on that.
Yeah.
I did I've submitted a bunch of stuff, but I have not worked on my CV at all.
Do you remember you were in the floor, right? 311.
Yeah.
When he came in.
Yeah.
Do you remember Dr. Pearson? Who was like, is there an administrator to do this?
Yes.
He's really funny. He's pretty knowledgeable. Oh, he emailed me the other day and he's like, can I register for this? I'm like, no, you didn't. And I'm like, and how's the CV coming along? And he's like, oh, you're that Aaron. I'm like, yeah. So it's just like, yeah. So just, it's sometimes hard for people to, when they, I don't know if we should just do stop share.
We did the assay.
Jason's not really talking.
We touted the assay.
I don't wanna.
And
I don't
they said,
wanna.
You'll do this assay.
I don't know.